Thank you, everyone. It's an honor to be here. John Bishai, Managing Director at Bank of America. I'm really honored to be here with this panel in particular because, to me, this is really the kind of heart of the industry. Behind the scenes, these are the players that are going to help advance these therapies, get them to market sooner. So this is an absolutely critical element of things. So without further ado, let me just introduce a few people. Mike Paglia, COO of ElevateBio. Chris Murphy, Vice President and General Manager of Vector Services at Thermo Fisher. Rahul Singhvi, National Resilience CEO. Ran Zhang, CEO of Landmark Bio. Thank you all for being here today with us. So one of the things I think about all the time as we think about manufacturing is there's been a stall. And one of the stalls has been last September with the FDA AdCom. And questions about how do we go ahead with manufacturing. I guess from your perspectives each, if we could go around the table, what are some of the lessons learned from that? I mean, thinking about the FDA and a decision like that, you usually anticipate a guidance, some sort of clarity, but there was no clarity. So we're going to look to you for a little bit of insights from that. Mike?
Thanks, John. Yeah, it's a great question because, a lot of companies like us at ElevateBio, we spend a lot of time actually looking at the guidances, providing comments on them as well. It's great having a CMC regulatory team that has experience and to be able to offer our partners, interpretation or help with interpretation of guidances, just like the recent CAR-T that was just published, as well as some of the gene editing. But that situation in particular, I think, we looked at it, we listened, and we circled around as a company and tried to look at really how can we help our partners understand that. Like, as you said, there is no specific guidance, but what we took out of it as a very heavily technology process development company was that, the characterization was key. So the process understanding that these products require, it's no, secret that the tools that we have or we had previously were not necessarily adequate to be able to characterize those products, understand the critical quality attributes, but really look from how are they performing in patients and making sure that the tools get better, that the processes get better, and that the process understanding gets better for these types of products in the gene therapy environment. So, again, that's what really we took out of that was we need to look at the technology that's out there, incorporate that, and really very early on look at not only how the products are performing, but also the materials that go into that process and how the outcomes are looking in terms of the characterization of those products when they get to the patients. So that was one of the major findings for us. And again, we help our partners and give that type of guidance on the bar is now higher to make sure these products are safe.
John, great to connect again. Great to be with this fantastic panel. When I think about it, really in the last 5 years, this technology has just exploded. It's been, I was working on it in the late 90s, and it was kind of moving along for many years, and there was this convergence of this great vectorology with clinical experience that resulted in Luxturna and Zolgensma being approved. But really, if you look at the data over the last few years, there's been a number of issues around clinical holds, safety questions. We're expanding our knowledge, we're expanding our experience. And I think the FDA, understandably, is proceeding with caution. These are long-term therapies. They're trying to understand the impact of these complex manufacturing processes. They involve plasmids, cell lines, different technologies. And I just think they're doing their job in terms of ensuring that the impurities aren't going to impact patients that are already very sick. So, I do appreciate the level of attention that's being paid here. And it does come down to, as Mike was sharing, expertise and really knowledge. You cannot rush the CMC portion of this technology. It takes years to get there and gain an understanding. And I think when we think about it in our business is really how do we continue to differentiate our analytical capabilities, our process development capabilities, because really, in the end, process is product. And I think that is where we're going to see some standardization, I think, in the next few years. But there's a bit of a, I think, as John pointed out, a bit of a pause almost here as we're looking at some of the data coming in from the clinic.
Anybody else have thoughts?
Well, I think the industry has made tremendous progress in the past few years with the first generation of cell and gene. Therapies are already in the market, and the second generation of the cell and gene therapy technology is already in clinic and demonstrate some very promising signs over there. And one of the biggest challenges, again, is in the CMC section. So when I think about the gene and cell therapy, and I think about a class of very diverse and complex medicine that we have never dealt before. And I agree with other panelists that understanding the product is the foundation of developing an effective processes, establish robust control strategy, and be able to reproduce or produce the products at a scale under CGMP reliably. And that's the fundamental learning that I have seen in the past few years. And one thing that talking about the complexity, that we have a complex product, we also have a very complex processes. Many cell therapy products now involve gene editing, and sometimes more than once and twice gene edits. And we're talking about iPSCs involve differentiation, so the processes are very complex. And then thinking about the starting critical materials still, that each of the materials, thinking about the gene editing, there itself is a biologic product. We're talking about plasmid DNA, we're talking about viral vectors, we're talking about the Cas9 proteins, and so that itself leads to the complexity of the supply chain. And I think right now that the best way is that how we can engineer out those complexities. While we're trying to figure it out, the first step is that we need to deal the complexity that we have to live right now, and how to manage those complexities given the supply chain aspect of it. And I would say that first map out your supply chain, find a partner that you can count on to make your products, and secure your trusted suppliers, and those are the fundamentals and the basics.
Yeah, I'll just add that in addition to what has already been said, that we have to characterize these products better. We have to ensure that we answer the question that the FDA is asking, is like, what is causing these safety issues? It comes down to having better characterization of what is going in, contaminants, et cetera. So completely agree. But the only additional thing I would say is that there is an incentive issue here with the biotech companies trying to get into the clinic as quickly as possible, because that's where value is created, right? So we cannot get away from that economic incentive. The model that we are trying to build resilience is to be able to do that in parallel. So can you sort of decouple the product development from technology development? Can we do the technology development so that we can give our biological innovators the opportunity to stand on stronger ground because the work has been done up front, rather than continue to propagate a situation where everybody is trying to do the same thing again and again? That's the only thing I'll offer, is that we need to decouple technology development with product development so that we have a better opportunity to scale this amazing biology that is unraveling in front of us.
And I think that's a great point, Rahul, because when you think about it, right, we're somewhat of victims of our own success,
Yeah.
right? You had Avexis, lights out data with the SMA. You also have Iovance, right, with the TIL therapy, as Pablo alluded to just on the last panel. But when you have these innovative technologies and we're rushing through the development pathway and they're getting expedited reviews, we're almost a victim of this and say, well, how do we supply this at a scale, right? So that interface of PD, which is very academic thus far, is now we're being asked to scale up at GMP. So how do we solve this? Chris?
Well, I mean, I think it's a great question. And I think, again, we talked about characterization. When I think about, how do we expedite really the PD portion, for me, a big part of this is interesting. When I came back, I was doing recombinant protein work up until about 5 years ago. And when I got back into gene therapy, I was amazed to see the technology hadn't really gone anywhere in about 15, 20 years. Now, no offense to anyone who's developed stuff, but what I'm trying to make the point about it is the scalability of this technology, the ability to get it to more patients, because we're going to see an expansion of where this technology is used. It won't just be for ultra-rare disorders and small patient populations. So I think a big thing for me on the development side is this engineering. And if you look at other technologies like recombinant proteins and monoclonal antibodies, really it's engineering that had the big breakthroughs on productivity and yield. And of course, you combine that with top-class analytics, particularly with these molecules, which are very hard to characterize. Potency assays are very tricky. I think these are areas that we need to invest more in, but it's costly. It's costly to do at-scale runs with the cost of these materials, these reagents, plasmids, you name it. But there is some concept there for me where we can learn from each other more and more, and I do think as a community working on this technology, we will start to share more and more of these learnings, and I think that'll be an opportunity to expedite development.
Maybe I'll add to what Chris just said. Again, going back to this point that when we look at these important biological innovations in cell therapy or gene therapy, many times, at least I'm observing, and by the way, I've been in this business for 27 years, but I've really only gotten into cell and gene therapy in the last 2 years. Before that, I was in vaccines. Interesting thing is a lot of things that I learned in vaccines are applicable to making viruses. So that's the good thing. The bad thing is that I don't know the history of this. But when I look back on the history of what has happened in cell therapy and gene therapy is that the biological innovation has been so strong that people have taken these very early academic processes and put them in factories. That has not changed because people are so excited about the results of the clinical data that they just want to rush quickly. There has been a general underinvestment in the area of manufacturing technology, in the area of really industrialization of this. And so we have an opportunity here to kind of go back and say, like, look, is that fundamentally the right way? Is triple transfection the right way to make a viral vector? Or can we do a better job? Is, like the previous speaker in the previous panel, Kristen from Bristol Myers was talking about how we can improve autologous cell therapy by engineering. Who's doing all that work? Who's going to front invest and do that? Is one company going to do that? Or can a company like ours can do it and do it horizontally, scale it? So I think that is the fundamental question is like, who's going to take the risk of really coming up with better platforms that will really scale these and improve the impact of these type of therapies for patients?
I think to add to that, there's still the innovation that's coming out of small biotech and academia, and what to Chris's point also, there's also not necessarily a platform for everything. Everyone loves to be able to say we have a platform approach, but having worked on these types of products, everyone is unique. Everyone has distinction. Everyone, especially from a potency perspective, is different. So tying back to the analytics and understanding how the product, and that's also something that often lags behind as companies come to work with us. They're often, okay, we're really good at process development and scaling into getting into GMP, but the analytics are immature and you have to work on those first. But oftentimes there's not a set of controls or standards that you can actually make development decisions. So a lot of the analytics has to happen up front. So I think, again, just to add to Chris's point, as the industry starts to harmonize and work together to come up with these controls, maybe there can be more of a platform process understanding, but it really comes down to harmonizing some of the analytics and the development of those assays so we can all make the same decisions and help to democratize what we're building for all of these different partners across the industry.
I guess that's the key question, right? We're all looking for a standardization of this, right, on the analytical side. So how does this come to be? I mean, given the fact that unlike recombinant proteins, antibodies and such, it's kind of the same format. These are very specialized products coming from different avenues, whether it be gene or cell. So how do we get to that point of standardization? Is it even possible?
Yeah, that's a very interesting question. Gene and cell therapy itself is a class of medicines that has many new modalities. And as much as we would like to see a homogeneous pathway for standardized processes, and in fact, each modality is going to be different. That said, I think within each modality, there are so many developments in science and engineering and technology front, and sometimes some technologies will emerge, and sometimes some technology will fail. But ultimately, the technologies demonstrate the most robust performance and gather most of the clinical experience will eventually emerge, similar to what we have seen in monoclonal antibody development. Protein A as a capture column, as one of the examples. So I believe that some of the technologies will eventually emerge within the modality of different cell and gene therapies. Now, across different modalities with gene and cell therapies, and I think there are some common denominators. Many of you touched on analytical method and analytical instrument, and those are the capabilities and technologies applied to any single modalities. There has to be an opportunity to standardize and to platform those technologies. And then another area that I think is related to gene transfer. There's a viral-based gene transfer and non-viral-based gene transfer, and those are the capabilities that you need to develop, whether you're doing iPSC, engineered iPSCs or CAR T cells. And then last but not least, the formulation is how you deliver, how you formulate your drug product. And those are the areas that I think they are the common denominators that the industry and academia can collaborate and work together.
Yeah, it was interesting. About 20 odd years ago, there was an effort to standardize, create a reference standard for adenovirus and AAV. It looks like Mike remembers that. And what's happened is that the whole technology now has shifted all these pseudotypes and these ways of getting vectors across the blood-brain barrier and things like that. So that's actually not probably where we're going to see the standardization. I think it's probably more on the impurity measures. Mm-hmm. Like how do you measure a full capsid? Because some of these potency assays you can imagine are extremely complex. How do you measure potency of a gene editing vector? How do you have a solid? So I do think we've got to start to at least standardize on probably some of the impurity measures at the beginning, and then because I think the potency and some of those strength measures may be a little harder to get there.
Yeah. Even the devices, just the analytical devices that are now evolving and coming out of new technology companies. It's exciting. I feel like it seems like weekly my AD department comes, Mike, we have to buy this. This is the best thing for impurity analysis. And empty and full are like, okay, what about the one we bought last week? But that's great. That's where the industry is really, the technology and especially on the high throughput automation, making sure that you're thinking about scalability commercially. And, the PD groups can just generate so many samples now that the QC and the analytics becomes, you can generate all the process understanding with these devices, but then that becomes a bottleneck. And having now high throughput technology that you can actually analyze all of the data that you're getting and make sense of it. The teams are just so fantastic across many companies on the way they're approaching DOE studies and generating samples for process understanding. So the analytics is now starting to evolve and catch up as well from a device perspective.
So going back to your question about standardization and whether it is even possible, if I go back and look at the history of what made the monoclonal antibody processes more standard, the industry actually worked together to standardize it, largely because of an economic reason, because they wanted to use each other's facilities. Amgen wanted to use Genentech's facilities, vice versa, if you remember from the old days. Pat Yang, my co-founder of Resilience, tells me the story that's what forced people to work together and create standard processes, because people had built their own capacities which were lying idle, and then a different company wanted to use their facilities. So that forced the standardization. I don't think that we have the time to do this because it may take 10, 15 years if that's the route you want to take. So our bet is that the model has to change. We have 800 public companies in biotech right now. So the number of companies are exploding because biology is exploding. Every one of them wants to create a bespoke process. So if we continue to propagate this, standardization is not going to happen unless there's a force that changes it. And the force in our view is that you have to go into a horizontal scaling company like the one that we're building, where we are trying to standardize things so that we give economic benefit to every company that says that if we sit on that particular platform, a standard platform, we'll benefit from it. So that ultimately, somebody has to do the upfront work to create a standard process so that others can bet on it. Say like, okay, I can go down a bespoke process and propagate this cottage industry, or I can actually work with somebody who can make a standard process and get benefit from that. So we'll see, how it plays out. But somebody has to put in the effort, the money, to create that counterforce to where the biology is taking us.
So I thought it's a very interesting when we're talking about the standardization, and now I'm thinking about with the diversity of cell and gene therapy, and many of them are still very early and emerging technologies. And what is the timing to standardize some of the technologies, and how we gonna do it? I think the first step is that we need to take those technologies, test it in human, and demonstrate the therapeutic potential, and prove that is a powerful platform where the industry and that can use. That is a very, very first step. Landmark Bio, we really very much focus on the translational space to take some of the early innovations from bench to clinics, and I think that's the very first step. We have to say, okay, which one is the winner, and so I think that's very important question, first step to answer. And once those winners emerge, and I think that's where the engineering, the technology is going to come in and try to standardize. So not just working on what are the platforms that work for now and for some of the first generation of gene and cell therapies that are already on the market, but thinking about what about the platforms of tomorrow, how that's going to look like, what the modalities this platform can support.
Yeah. No, that's very helpful. And I think, Rahul, you also touched on something, right, the consolidation aspect of it. And we're kind of in the midst of this now with the capital markets hitting a bit of a downturn, especially in the cell and gene therapy space, with a number of companies down over 50% on the year, which is surprising, right? Previous years, for the last few years, you've been able to be a cell and gene therapy company, go to the capital markets, raise considerable capital, and even take on manufacturing on yourself. Whether you have the expertise or not, that's another thing, and we could talk about that. But the markets allowed you to go ahead and pursue those streams and scale. Now we're seeing, when I first started in this sector, I was close to Brammer with you, Chris, represented Thermo on that acquisition. Then we saw Paragon and Catalent. And then shortly after, we've seen a shift, right? We've seen Atara sell their facility to Fuji. We've now also seen Homology Medicines pair up with Oxford Biomedica to create a unique supplier, if you will. Like, with the capital markets, how do you guys view this, right? Does this mean more consolidation for you, to your point, Rahul? Is this where this is going?
Yeah, I mean, again, the markets are telling them become more capital efficient. This whole idea of bespoke processes, in the rubric of I want to control my own destiny, is going to lead to a lot of inefficiency. I understand why people do this, because there's not really good service on the other side, apparently, and so they need more reliable, trustworthy service so that they can rely on the third party to do this. But if a company is able to do that, and they have that kind of track record, then the markets are pushing them to go in that direction and avoid the duplication of work that is currently going on. Many of these companies are running manufacturing without the volumes, and so they become cash burning engines. And so in a time frame when capital is scarce and stocks are down 60-70% from the IPO prices, first thing they need to do is sell that dog.
Yeah.
Oh, well, just it's interesting. I think Rahul's point is so spot on. Like the touches, the volume of throughput is so crucial in this business. This is very tough technology to master, and if you're not routinely running these processes, you have challenges, you have rework, you have a number of issues there, and of course, very expensive facilities to operate. I mean, single pass air in most of our facilities. That's brutally expensive on utilities, particularly recently. And so I do think that, and then when you look at the attrition rate on these molecules, isn't that different than what you're going to see with other technologies. I think if you go back 5 years ago, it felt like every molecule was going to win. you know, and I think we're seeing that that's probably not going to be the case, unfortunately, especially for the people that need these medicines. But the reality is, I do think you're going to see the folks that can differentiate the capabilities, have enough volume, have a workforce that's touching these processes. And to Mike's point earlier, you know, we don't have one platform we operate. There's multiple platforms we use. So I think you are going to see probably more of a shift probably to where there could be some consolidation of CapEx and our capital facilities, and then reliance on people with the expertise.
Yeah, when we, you know, with Elevate being about 4 years old now, our founders saw this coming almost, where, you know, David, Vikas, and especially Mitch, having built, you know, 5 to 10 cell and gene therapy companies over his career, you know, it was just time and time again, it's the talent question as well. Getting the expertise, hiring the staff that has the education, the know-how, not only to do GMP manufacturing and the engineering, which is important, but to develop those processes right from the beginning. The raw materials, the controls, again, the assays, the development itself. So we see that in these companies as well. They have great talent, but there's something always lacking, whether it's the analytics, the development, or the GMP know-how. So I think with this market currently, you know, setting up a model where you can get access to that talent, that those folks that have worked on approved cell and gene therapy products, that know the regulatory environment, that even know the clinical design or the immunotherapy, the preclinical work, getting access to all of that all under one roof at different companies, it's a benefit for a biotech company to take that approach, and especially in this environment right now, because you really can't compete on the talent. You know, hiring the folks, it's a little scary out there right now to go to a startup company or a startup biotech in this environment. And, you know, again, people do, and you want to work on the innovation, but to de-risk it, especially the CMC, with the models that companies like ours have set up, it seems to be very attractive in this environment right now for companies to really help to develop those therapies rapidly.
Market condition is one factor. Even when the market was going very well, and we have seen a lot of innovations occur in the academic setting for young startups and may have difficulties to access capital even when they are able to access capital, but the inability to stand up their own facility and hire the talent has been a challenging situation for many of young companies and life sciences innovators in the academic setting. So I think the key question is that how we can solve that problem regardless of whether the market condition looks like. And I think the answer is that we need to partner with the innovator companies and academic researchers to find a way to help them to move their research from bench to clinics in a capital efficient way. And also help to de-risk in general for the entire industry to going through this cycle to build and then close down and divest. And this is a vicious cycle.
I guess from my perspective, one of the things that came up in the panel here today was tools, right? Like the reality is a biotech company, we've seen a slew of companies focused on AAVs. Now we're talking about next generation AAVs. We've talked about Lenti, but now we're seeing a slew of companies come with different types of editing technologies. There's a new trick every time, right? New tool. How do folks like you start thinking about these tools? Should you be tool aggregators? Should you guys master the tool and out-license this? Like, how does this start changing your perspective of the tool portion of it? Because, again, that's just one aspect of the therapeutic.
I mean, for us at Elevate, the answer is yes. I think, you know, we thought about it from the beginning. You know, what do you need to build from the beginning to enable a therapeutic in cell and gene therapy, and where's it going? We have gene editing technology, our company LifeEdit, and we have inducible pluripotent stem cell lines, you know, that we built very early on, GMP compliant, ethically derived, everything that was lacking in the regulatory environment to be able to actually innovate on the cell therapy side, but also on the gene therapy side. And as you said, you know, next generation ways to edit cells or deliver the genes is something that's important. So having all of that technology, whether it's an LNP or an AAV, and offering that, and again, I think the word that we've used a couple times, democratizing that to be able to enable the industry, so to provide those tools to partners is very important. And again, that know-how, that regulatory experience to help them de-risk, I think it's a great approach. And again, we're not the only ones doing it, but seeing that vision of where the industry is going and building that technology to help enable, it's something that is very exciting, and to be able to offer partners, and they see the value in that as well.
Yeah, I think, I was going to say that, I completely agree with Mike saying, and from my view, if you just listen to your customers, listen to the regulators, and of course the patients that our customers serve, speed is important. You don't want to have a big, a complete response letter when you're submitting it. You want to have full characterization, all that. And these are complex processes. It used to be you'd have a producer cell line alone, and that would secrete your product. And actually, that was still very complex, of course, for those of you that worked on that. Now you've got plasmids coming into play. In some cases, if you're using a baculovirus system, you have viral banks, things like that. So I do think if we can find ways to recognize that the single-use reactors we use, the media we use, the cell lines we use, the plasmid, if we understand those and can pull them together in a way that can help expedite getting that first patient into your trial, I think that's hugely valuable. And of course, if you can do it in a way that has a very good touch and understanding of what the regulators are seeing is very important, I think that's a win for everybody. It's a win for patients, it's a win for customers, a win for our industry. So that's where I see the value really of aggregating these tools.
Yeah, I agree with both Mike and Chris. We do serve a purpose of aggregating the tools. In addition to what they said, I would add that IP is another consideration. In many cases, these tools are held by certain universities or certain companies, and the rates, it's difficult to navigate through the whole licensing process. Somebody, if they can serve the customers, in this case, the biological innovators, in helping them through that so that they don't have to go through the licensing, that would be service. That's one point. Second point is, like, many times you take the route of least resistance and go after a set of technologies that are easiest to license or are easiest to grab, but that may not be the right solution for your product. In which case, for either ease or economic reasons, because of royalty stacks, you're picking the wrong things and pairing them with the product, which is not ideal for ultimately patient impact. So one of the things, these are the considerations that I think that when I think about tool aggregation is, in addition to make it easier, can we provide a choice to the customer where they have the ability to pair the right technology with the product without the adverse effects of royalty stacks and so on, because once they know what technologies will work, then they can deal with the economics later. So that's a service that a company like ours can provide.
So when Landmark Bio was first still at the conceptual stage, we thought about the diversity of this field and the different modalities and the technologies that we need to pursue and or acquire or develop. And we started with our facility, and we want our facility to be modular, flexible, and can be easily changed to different technologies, because this field is still growing, and new technologies coming out every day. And now, with our facilities are pretty much built, and we're taking in some of the most innovative early stage projects, we start thinking about how we could incorporate innovations into those therapeutics, and first enabling them to get to clinics and being tested in human. Second, and to develop the technologies and manufacturing technologies or analytical tools to enable those, because many of those novel modalities don't have a path to follow to manufacture. So this is where we believe we could contribute and help and make a meaningful difference over there. And then through our network and industry collaboration, as well as academic collaboration, there are a number of technologies, solutions, toolbox being developed, and this is where we could evaluate and de-risk and bring them in. And ultimately, the goal is that to create a platform for tomorrow's gene and the cell therapies to bring them together. So I think one key point is that when we think about those new tools and technologies and figure out how to integrate the innovations into therapeutic development, it's going to be key.
I guess, in the last few minutes that we have, I'm just going to put myself in, say, a biotech company's position, right? If I'm a private biotech, limited capital, what's your piece of advice for them at this juncture, given the capital markets being where they are? I mean, is it, hey, come talk to us as soon as possible? Is it, hey, just don't rush through with an academic product? Like, there's got to be some lessons learned over the last few years that there's got to be something that we can grab onto from the last few years. Rahul.
So I think one is that set expectations with your investors so that they don't have the wrong view that you can get into the clinic within a year when you don't have a process or you don't have the CMC worked out. I think this is I know in the enthusiasm of building a company, we tend to make these type of promises largely because we don't understand what it takes. And it leads to disappointing the investors. It leads to sometimes misleading them, and it doesn't land well. So understand what it takes to do this. I would like to think that if today or in the future we would like to be a company that they can work with, that can carry the water on this end, while they prosecute their preclinical and clinical programs.
Yeah.
But I'm sure like my colleagues here have the similar value propositions. But the idea is that set expectations so that you don't disappoint your investors.
A lot of our partners come to us already thinking about the third generation product. Already where it's going to innovate to. They know they need first generation, like we've all started in the cell and gene therapy. You have to get into that first generation. But they're even, from a regulatory perspective, getting more complex. There's combinations. It's not just a single modality anymore. So, we like them to focus on the innovation side, making sure the preclinical packages and take some of the heavy lift of the CMC off of them. Understanding, like, we've done this before, we can help you with the CMC. It's complex, but we understand it, and we can help you understand it and take that path together. But actually use your resources on your next generation. You have great scientists, great innovation. You're going to develop new therapies and additional modalities and new products. Focus your time there, and we can help you de-risk that manufacturing in the CMC. So that's kind of how we work with partners, and they get excited about that because they do often come out of academia and they want to work on next generation or taking that product into multiple clinical indications, which is complex.
Yeah, I think it's to me it's like you have to know what you're good at and what you need to be good at. And I think that's important because I think it's hard to be really good at everything, and you have companies that are often started because they're very committed and very passionate about the patients they serve. Have so much respect for that. But it doesn't mean you're an expert in every element of what it takes to make these products. And I don't say that because I'm not an expert in the clinical side, but I can tell you that we work often with small companies that want to be the expert in everything, and I think that is a tough thing to do. It's just tough to do, and you have to prioritize. You really have to prioritize what you need to be good at in the context of, and this is where it is a partnership. It's a partnership with a CDMO, a supplier, with a clinic, however you want to look at it, but I think that's super important.
I would add to my panelists' comments, and I would say first to have a long view. Drug development is a journey, and it's very important to have the end game in mind. So do not accelerate a particular step and then slow down the entire process. We have seen this before, and we have seen this happening again nowadays. So it's really important to map out your entire drug development journey and figure out what is the best path to get to your drug eventually, prove in clinics and launched in commercial. So that's number one. Number two is use your capital judiciously. A lot of companies raise a large sum of the capitals, and there's great excitement over there, and do not lose your focus, and make sure that you allocate the capital in the areas that can give you the most, advantage moving forward. The third one is that if you do decided to partner, find a true partner, and find someone that in the CMC space that can help you navigate through the complexity of the CMC development and regulatory path. And that is really, really important, especially for the early stage innovators and companies.
Well, looks like we're out of time. I just want to say thank you to all of you. You've really helped enlighten us a little bit more on the manufacturing side. And I guess right now we're going to take a short break, and we'll return at 3:40.